First of all, thanks a lot to Jerry Morgan for arranging all the logistics for my visit here. That is a big job. And thanks to Larissa for serving as my host. So the email I got from Larissa inviting me to do this started out with the line that said, "Dear Winsor, I hope you're doing well, and I hope this email brings you some joy." And it did bring me joy. It brought me joy because how often do you get an email that says, "I hope this brings you joy"? I don't get very many, and the ones that do bring me joy, it's really hard to tell. So that was really nice, and it does bring me absolute joy to be here. Well, I should say joy with a mix of terror in there, to be here at a place with such a high concentration of scientists who I admire, and such an amazing record of producing excellent graduate students. So it is with that mix of joy and terror that I am here. All right, so when I got into my PhD research was the time when the metapopulation world was going nuts. So Ilke Hansky and friends were pumping out paper after paper in Science and Nature showing that dispersal has all these really strong effects on everything we care about, so population ecology, evolution, community interactions. And so the overall message from that research, and what got me super excited as an ecologist, was that dispersal is this really important process. But the catch with dispersal is that it's often happening at really large spatial scales and in really complex landscapes. So it makes it really hard to quantify directly. So we know that it's important in things like evolution, for influencing gene flow among local populations, genetic diversity within populations, and important things like adaptive potential. We know it's important for population biology and demography, for rescuing declining populations or maintaining metapopulations in these complex landscapes. And now, with this explosion of meta-community theory, we know even more how important it is in maintaining diverse communities and biodiversity on the landscape. But what it took me a little while to figure out is that most of what we know about dispersal comes from indirect inference, so indirect data. Things like patterns of genetic divergence among populations as quantified by FST values, or spatial and temporal variation in local population abundance or population occurrence, or spatial and temporal patterns of species co-occurrence in patchy populations. So from those kind of data, we would infer the role of dispersal. Now those indirect indices of dispersal are really important for the reason that I already mentioned, because often dispersal is happening at this large spatial scale and in complex landscapes, and quantifying it directly is difficult or impossible. But my agenda today is to try to convince you that in systems where it's feasible, a more bottom-up approach to studying dispersal may be valuable. And by that, what I mean is starting with direct data on individual variation in movement behavior, emergent patterns of dispersal at larger spatial scales, and then using those data, aggregating those data to characterize population-level distributions of things like dispersal propensity, so who stays, who leaves, or variation in dispersal distances. So why do I think that more humble kind of bottom-up approach is valuable? I think for two reasons. Number one, it gives us this empirical foundation for using and interpreting those indirect indices, so things like FST values or spatial and temporal patterns of population occurrence or abundance, in a more scientifically sound way. So using those data, we can develop predictions about those broader consequences of dispersal, and then we can test them with the indirect indices. The second reason why I think it's important is that I believe that by starting at the individual level, so quantifying individual variation in dispersal propensity, dispersal distances, and most importantly, the fitness consequences of dispersal, that's the only way we're going to get beyond kind of cataloging these diverse effects of dispersal in different systems to, and this is a very biased opinion, to what I see as the real interesting question, which is how does dispersal evolve and how is it maintained in natural populations? So a more basic insight into this very important ecological and evolutionary process. So for the talk today, I've structured it around a study that I think highlights what we've learned about the causes or the drivers of variation in dispersal distances within populations, what drives long-distance dispersal events, which are really especially important both in the ecological and evolutionary context. And then I'll move on to another study that uses those direct data on variation in dispersal distances to infer something about broader patterns of gene flow and genetic divergence in complex landscapes. And in this case, it's going to be the complex landscapes of headwater stream networks and stream salamanders. So first, causes. And this is work that I did in collaboration with Mark McPeak, who's at Dartmouth College and who was a committee member of mine and has just been a collaborator and influential kind of mentor since then. And this is work looking at the drivers of variation in dispersal distance in natural populations. So what this is an empirical dispersal distribution for my study species, a stream salamander. I'll tell you more about that guy in a few minutes. But I'm going to take a minute to explain what this is showing you. So this comes from long-term mark recapture data in a single population of this species that's in a small stream in northern, very, very northern New Hampshire. And over 6 years, so I visited this stream and used consistent sampling methods to mark and recapture animals along this 1 kilometer long section. And this is the lifetime dispersal distribution of those recaptured animals. Here you have 0. So those are in the middle. So 0. So these are the guys that don't move. They're essentially the ones that I capture under the same rock almost every year. And then the positive values are upstream moves, and the negative values are downstream moves. And then on the y-axis, you have number of individuals. So this is just a frequency distribution of dispersal distances within this population. And what I want you to notice about it, which is pretty apparent, is that it's leptokurtic. Now that's probably not the word that jumps to your mind, but that describes this fat-tailed dispersal distribution that you see here. So a high peak at 0, the guys that don't go anywhere, and these fat tails that are comprised of the animals that go relatively long distances, up to 550 or so meters. Remember, this is a stream salamander, so little legs, 550 meters is pretty impressive, I think, for a salamander. What's interesting about this dispersal distribution is that we think that this characterizes the dispersal distribution of most species, both plants and animals, so this leptokurtic pattern with a lot of individuals that stay very close to where they're born or their origins, and then these long tails or this large variation in dispersal distances of the movers. So this seems to be a common pattern, if not universal pattern in nature. But the trick is, in most systems, it's really hard to quantify well because it's happening at these really large scales and in multiple dimensions. Whereas here, the nice thing about working in a stream salamander is it's just salamanders moving up and down an individual stream. So we can quantify that pretty well. And there's been this real surge of interest in those fat tails because it turns out, not surprising, that they matter a lot for things that we care about, such as the spread of invasive species. So those long distance dispersal events are especially important for the spread of invasive species. Range shifts in response to climate change. Turns out the fat tails matter. And then just simply maintaining viable populations in complex landscapes. It's really those long distance dispersal events that matter the most. And so I was inspired basically to ask, can I use this system and these empirical data on variation and dispersal distance in this humble stream salamander to try to understand something about what causes that variation in dispersal distances in a natural population. So hoping to provide broader insight on what appears to be a very common pattern across species. And this is the focal species. So this is Geronophilus porphyriticus, the northern spring salamander. It's pink, which is nice, and it's large for a plethodonid salamander. I should mention first, so this guy's a member of the family Plethodonidae, the lungless salamanders, which is the most diverse family of salamanders. A lot of that diversity is concentrated in the Appalachian Mountains. The northern spring salamander occurs from Alabama up to southern Quebec, and it's mainly found in really small headwater streams. So the streams I work on are usually no wider than this stairway here. It has strictly aquatic larvae, so they stay in the stream, they have external gills, and they stay there for about 3 to 5 years, and then they transform to become these semi-aquatic adults that hang out most of the time in the stream itself under rocks. But they can actually get up and move out into the riparian zone and forage at night, but then they tend to come back to the stream. So it's a very aquatic salamander overall, but it can make these forays into the terrestrial habitat as well. What else do you need to know? Long-lived, up to 14 years, we think, for adults, and a notorious predator of other salamanders, especially in the southern Appalachians, although up where I work in northern New Hampshire, they eat mostly aquatic insects. So it seems to be a difference there. And the data that I'll be showing you today comes from this long-term mark recapture study of Geronophilis in a stream called Merrill Brook, which is in, like I said, in northern New Hampshire, right up near the border with Canada. And the study design was really simple. So this is the main stem of the Dead Diamond River that's infested with brook trout. But there's there's a wetland there at the base of the stream that seems to block brook trout movement up into Merrill Brook. And then Merrill Brook runs for about 1 kilometer up a slope there. And I surveyed this entire 1 kilometer long section of the stream. And I do that repeatedly for 6 years, returning June, July, and August and marking all the animals that I encountered along that 1 kilometer long section, recording things like, well, their position along that section, as well as length, mass, I took photos for morphological analyses, but a lot of just individual base measurements along that section. So I end up with this spatially explicit data set on salamander distribution and movement within that 1 kilometer long section. And it's important also to note that that covered the majority of the perennial portion of Merrill Brook. So it goes dry up here, but this was the perennial portion. And I'd survey once in a while up here just to make sure I wasn't missing salamanders that were, leaving the perennial portion. And like I said, so I did this mark recapture study intensively for 6 years when I was out in New Hampshire. Since I've moved back to Montana, I only can get there once a year, so I've had to stop the actual marking, but I continue to return to Merrill Brook every year to just maintain that long-term data set. And I'll actually be talking about some of those data tomorrow in my talk. Okay, so basic question here is what's maintaining that variation in dispersal distances within this population of salamanders, and what can that tell us about these broader patterns across species? And what I won't be talking about today is that we know that environmental variation does contribute to that variation in dispersal distances, specifically variation in the timing of, or in stream hydrology over the course of the season. So as you can imagine, that influences what motivates a salamander to get up and move if the site that they're in is going dry, and that then plays out in influencing that variation in dispersal distances. But what I want to focus on today is the role that natural selection might play in maintaining that variation, because that, I think, is the key to understanding whether there's this broader there are these broader mechanisms responsible for this common pattern we see across species. So asking, does selection play any role in maintaining that variation in dispersal distances within this population? Since I'm going to be talking about the evolution of dispersal, I need to acknowledge that this is a question that is really hard to answer, and it's stumped much greater minds than mine, including Fred Allendorf, who's my great friend and mentor at University of Montana and who I have lunch with every week, and who said one time, thinking about the evolution of dispersal makes my brain hurt. And then one of my grad students, Brett Addis, found this great quote from Ernst Mayr, who I think was also struggling with this question. So the question of how does dispersal evolve, but also how on earth do we test that empirically in natural systems. And so I'm just going to read it. So too much dispersion destroys ecotypic adaptation. Too little induces inbreeding. Again, the advantages of the proper balance seem to be for the population as a whole rather than for the individual. How then is this balance regulated by natural selection? And what I really like about this quote is that it reflects this common trap that I fall into, which is thinking about dispersal and its benefits at the population or even at the metapopulation. So what's the right balance of animals that move among populations versus those that stay where they're born? There's always that tendency to think about it that way, but we know, all of us in this room know, that you don't get anywhere thinking about evolution at the population level. You need to think about it at the level of the individual. So what are the fitness consequences for the individual? And what he gets to eventually is it would seem preferable to search for solutions based on the selective advantage of individual genotypes. So, all right, let's try not to think about it at the population level. Let's boil it down to the individual and see what's going on. Now, just to tamp down expectations, we are not here yet. Someday, I would love to know if there's a, genetic, or I would love to know the genetic basis of variation in dispersal propensity or distances. We're working on that question, but today what I'll show you is some data on the fitness consequences of variation in dispersal as one step towards that ultimate question. Okay, so when it comes to this question, what maintains that variation in dispersal distances in natural populations, I'm going to simplify the literature down here a lot and say that there are two main hypotheses, and one of them is pictured here, and that is that variation that we see, those fat tails, are explained primarily by chance and stochastic forces. And that's reflected in those citations that I have up there. And there is no doubt that there are many systems where that is absolutely true, that what causes variation in, say, wind-dispersed seeds, a lot of that variation is caused by just chance wind events that might pick up one seed and send it a long distance. Or another great example, these pelagic larvae of reef fishes that get picked up by a current and sent super long distances. And we've all heard the stories about rafts of coconuts carrying lizards between islands. And there's no doubt that those things happen. So one possibility is that it's all just chance. So the fact that we see this common leptokurtic pattern of those fat tails across systems is really spurious. It's not saying that there's a common mechanism or that there's natural selection plays any role in it. It's just saying, yep, chance happens all over the place and it causes that variation. Well, that leads to a pretty straightforward prediction. That is that if we looked at the relationship between dispersal distance then, here on the x-axis, and some measure of fitness here, we shouldn't see any predictable relationship. If it's all based on chance, there should be no deterministic, there should be no predictable consequence for fitness leading to plenty of scatter. Sure, there's a chance you're going to land in a good place, but no predictable fitness benefits of increasing dispersal distance. And here, as soon as possible, I'm also going to add this caveat here. When I talk about fitness here, I'm talking about measures of fitness that I can collect in the field. So I'm not talking about fitness with a capital F, as you'll see in a minute when I show you what I can actually measure in these salamanders. Okay, so what's the alternative hypothesis? Is that, yeah, maybe natural selection does play a role in maintaining that variation in dispersal ability or dispersal distances. But the trick here is that most of the evidence or the support for that hypothesis has come from modeling. It's come from theory. And there's a lot of really interesting papers suggesting that long distance dispersal can be selected for given in certain metapopulations with certain configurations of habitat or during range expansions. So, for example, what might be caused during climate change. But again, a lot of it comes from theory with very little empirical tests of the role of natural selection. But this leads to another straightforward prediction. So if natural selection does play a role in maintaining that variation in dispersal distance, if we plot, again, dispersal distance here on the x-axis, we would expect in some fitness measures to see a benefit, so to see a positive relationship between how far you can go and the fitness consequences. So we have this overall question. How is that variation in dispersal distance being maintained in natural populations? We have two relatively straightforward predictions. Either it's all by chance and stochastic forces, in which case we'd expect to see that flat line, or if natural selection does play a role, we might expect a positive relationship between distance and fitness measures. And here again, I think it's important to point out that this would kind of keep the door open on the role of natural selection in maintaining that variation, but it certainly wouldn't prove absolutely that that's the case. We tried to test that using two overall approaches here. The first was to use those long-term mark recapture data to simply look at the relationship between dispersal distance of those marked and recaptured animals and fitness consequences. Specifically, survival probability and growth rate. And I've kind of alluded to this before, but a lot of what limits our understanding of, again, this role of selection and dispersal is the lack of these post-dispersal data. So being able to figure out what happens to individuals after they land, wherever they land. And again, in this humble example, a 1 kilometer long section of a headwater stream in northern New Hampshire, I was lucky to have those data for a relatively large number of salamanders. And what were the fitness measures that I could quantify there in that stream? First, survival probability, using those mark recapture data. So I mentioned this before, but Geronophilus is long-lived, up to 14 years, and annual reproduction is relatively low. So these are not the explosive breeders that you sometimes see among amphibians. For these guys, they appear to kind of trickle out low reproductive effort across their long lifespan. So suggesting that survival, lifetime survival, is probably an important component of fitness as opposed to reproduction in any particular year. Growth rate, that could be a good index of fitness because we know that in Geronophilus, there's a positive relationship between body size and the egg production. So the larger you are, the more eggs you produce. And that's based on some earlier data that another scientist had collected. So we have these two possible indices of fitness in the salamander that I can quantify in this field population. But again, we're not talking about lifetime fitness with a capital F here. It's just as close as I could get. So first, results for survival. So this comes from mark recapture data using program mark and Cormack-Jolly-Sieber models using dispersal distance as an individual covariate and basically asking, does knowing dispersal distance improve our estimates of individual survival, or does it add information to the model of individual survival? And it turns out that it does. So on the x-axis, you have dispersal distance. This is log transformed, and then this is relatively yearly survival on the y-axis. And what you can see is that there's this positive relationship. So the farther they go, the higher their survival is, although it does seem to asymptote out here. And I have a red circle around the 0 there to remind you that the animals that don't move are in here. So what this is saying is that even relative to the guys that just stay put, so stay under the same rock, the ones that disperse tend to experience higher lifetime survival. Now, a question that often comes up with this graph, and so I'll try to head it off here, is that, well, couldn't you get that relationship if you just ended up recapturing these dispersers more often than you recaptured these guys down at the 0 end? And it turns out that there's no relationship between capture probability and dispersal distance. So there doesn't seem to be a bias caused by who I was recapturing more often. It appears to be this real relationship with survival. Okay, so how about for growth rate? Now, this is a pretty ugly relationship here. Here you have dispersal distance on the x-axis, and then proportional daily growth rate on the y-axis, same coming from these recaptured animals, so I could measure them, their size when I first caught them, and then after some interval, I can standardize that to a daily growth rate. And then look at the relationship between dispersal distance and growth rate, and you find that there's a positive relationship. So despite all this noise here, the trend reflects an almost threefold increase in growth rate across that range of dispersal distances. However, it's obvious you would never want to have to predict growth rate based on dispersal distance because there's a lot of other causes of variation there. Okay, so going back to the big question and predictions here then, we have based on 2 measures of fitness that I could collect for this salamander, we have some evidence that selection may be playing a role in maintaining that variation in dispersal distance. So there do appear to be fitness benefits of increasing dispersal distance in this species, which, like I said before, this is a difficult question to get at, and so I take this kind of with a grain of salt, but it keeps that door open that, well, maybe this pattern that we see across species is not just about chance and stochasticity, it may be selection is playing a role. The second way we tested these predictions was to look to ask whether there were phenotypic correlates of dispersal distance in this species, and this is kind of a natural next step. So if you see fitness, that there are predictable fitness benefits, then you might expect there also to be selection on phenotypes that would promote variation or promote long distance dispersal. And to do this, so leads to almost the same prediction. So you can look at this phenotypic trait. If it's about chance and stochasticity, we wouldn't expect to see any correlation between the trait and the dispersal distance if it's about chance. Whereas if if selection is acting on the phenotype and promoting these phenotypes that allow for long distance dispersal, you might expect to see a relationship between the trait value and dispersal distance. And there's no reason that this would have to be positive here. You just expect some relationship between distance and whatever your continuous trait that you're measuring is. Okay, so to do this, went back. Thankfully, I took some photos of some of these marked and recaptured salamanders over the course of those 6 years. So I could go back to those photos and do simple morphological measurements using those photos. And we measured head morphology, trunk morphology, and leg morphology based on those photos. And then size corrected them so that it's not so it's not biased by variation in the size of the individual salamander. You have these size-corrected morphological traits. And then ask whether any of those traits show a relationship with dispersal distance. And I guess surprisingly, one did. Not surprisingly, it was the legs, because that you use those for dispersing. And don't worry too much about this. The direction of the trend here, it's just based on how this leg principal component was calculated. But what you see is that, so you have dispersal distance here along the y-axis, and this leg principal component, which again is size corrected on the x-axis. And what you can see is that animals that had relatively long forelimbs and short hind limbs tended to disperse the farthest, and animals which showed the opposite kind of leg morphology, so relatively long hind limbs and short forelimbs, dispersed shorter. So there is this evidence of a relationship with morphology, and actually there have been other examples of this kind of relationship in other amphibians, including in toads in Australia, perhaps most famously, showing that the toads with the longest legs are often at the leading edge of the invasion for the cane toads in Australia. But again, kind of surprising to find this in this one small stream in New Hampshire. Overall then, based on the measures of fitness and then the measures of leg morphology, we have more evidence that selection may be playing a role in maintaining that continuous variation in dispersal distance that we had seen in this population. I should have also mentioned this before, but if you think back to that dispersal distribution, we know that that's consistent both for the larvae and the adults, and it's consistent for both sexes, so for the females and the males. So it's not about kind of the females are the ones going the long distance, the males are not, or the larvae are the ones going the short distance and the adults are going long distances. It seems like everybody is kind of is showing the same pattern of variation in dispersal distances. For me, again, what's exciting here is that it's, again, scratching the surface of what I think of as a really exciting and important question, which is how is this variation in dispersal distances maintained in natural populations, and does natural selection play a role in it? And I'm always very aware that there are so many other aspects to this question, and it's hard to get at, but I think it does show the value of those direct data. So actually going out and measuring variation in dispersal distance for testing that question. So natural selection does appear, based on these results, to be playing a role in the frequency of those long-distance dispersal events. For me, the important next questions is really looking at how that environmental variation may influence how selection is imposed on variation in dispersal distance. So when you start to add things like variation in stream morphology or stream temperature or physical structure of the habitat, does that change the selective regime that's experienced by the population and change the patterns of dispersal that we see within those populations? I think this is, again, it's really important for just testing the generality of the results from this one stream in northern New Hampshire. But it's also really important for thinking about how things like climate change may influence variation in dispersal or influence selection on dispersal strategies into the future, because it's going to be adding a lot of that environmental variation. To get at that, so to get at this environment by evolution interaction, what we're doing is recreating that same study. So the same basic design, walking streams, turning over rocks, marking salamanders, returning, marking more salamanders year after year. We're recreating this at Hubbard Brook, which is a site in central New Hampshire where there are lots of salamanders, so check, that's important. There are lots of streams, what's figured here. So this is the main stem of Hubbard Brook here, and then you have all these tributaries coming off of the main stem, and they're loaded with salamanders. So we got streams, we got salamanders, and then it's Hubbard Brook, which some people may be familiar with, but this is a place that's known for research on ecosystem ecology, basic stream biogeochemistry, kind of stream terrestrial interactions. And that's just fantastic, because what that means is that we have a load of data to add to our simple mark recapture methods and ask, how is the environment mediating these patterns of dispersal or just basic demography that we see in these stream salamander populations? And this photo just shows a weir. So these are all the streams that we have pictured here. And there are 5 streams where we're doing this, have weirs on them where they have conducted long-term stream chemistry analysis, which again is great. The only reason we can do this salamander work at Hubbard Brook is because we work below the weirs. So you can't do anything above the weirs, because then you're going to mess with their long-term stream chemistry data. But we can do whatever we want below the weirs, because they're not worried about that. And I have a grad student who's working on that project. Okay, so now moving on to the next part of the talk, so talking a little bit about the consequences. So in the first part, what I hope to show is that we can use these direct individual level data on dispersal to try to understand something about the evolutionary basis of that variation in dispersal distances that we seem to see across species. What I want to show you now is a study that I think illustrates kind of the next level. So taking those data and trying to understand landscape level patterns of the consequences of dispersal. And in this case, it's mainly going to be looking at variation in genetic divergence along headwater streams. And again, this is work that I did in collaboration with Mark McPeak, Brad Cosentino, and Gene Likens. Gene Likens is my main connection at Hubbard Brook. So he's basically started Hubbard Brook and is a world-renowned biogeochemist, but he loves salamanders. So it's really fun to work with him there, because it's kind of like, for me, what's my work is his play, and for his work, he thinks about nitrogen and phosphorus. Okay, so I mentioned before, Geronophilus is in the family Plethodontidae. The Plethodontidae are the lungless salamanders, and they rely on cutaneous respiration, and they're super diverse. And here I have to go to my notes just because I will get the numbers wrong. So they're the most diverse family of salamanders, with approximately 340 species. And a lot of that diversity is concentrated in North America, which you can't say for very many groups of species, particularly along the Appalachians. So super diverse. A lot of the species are associated with headwater systems. And like I just said, so a lot of that diversity is found in upland headwater areas, along small streams and in the riparian zones adjacent to those streams. And that's true in the northern Appalachians, or throughout the Appalachians, I should say here, but also in the second big hotspot of plethodontid diversity, which is Central America. So you see a lot of diversity down there, and where most diversity is a subject of heated debate within the salamander community, but I'm with Team Appalachia. Okay, so have this high diversity, and we have a lot, and because of a lot of that diversity is here at home in North America, the plethodonid salamanders have been an important study system for understanding patterns of species diversification. With a lot of great work, both in the Appalachians and in California. What we asked with this study is there something about these habitats, these upland headwater systems, that may promote genetic divergence and diversification within this group of salamanders? Something about that steep topography, for example. And we came up with this super obvious hypothesis that maybe there's something about the topography, the steep topography of those headwater systems, interacting with the dispersal patterns of these species that, again, promotes divergence within populations and then deeper levels of divergence at the species level over time, although I'm not working at those levels. The trick is we needed to know something about dispersal in these species to think about this interaction. And in the case of Geronopholis, again, I did know something about dispersal based on these long-term mark recapture data. So does that interaction promote divergence? This is the same figure I showed you earlier. So this is that dispersal distribution for Marlbrook up in northern New Hampshire. And we already talked about the fact that it's leptokurtic with that high peak in those fat tails. But the other big characteristic of this distribution is that it shows a strong upstream bias. What does that mean? So most salamanders don't go anywhere. Just like one of my committee members said when I started working on salamanders, why would you want to study dispersal in those salamanders? They don't move from the same rock for their whole lifetime. And strangely enough, that's absolutely true for most individuals. But those that do go somewhere tend to move upstream. And I mentioned this before, but remember, seems to be true for the adults and for the larvae, and it seems to be true for both the males and the females. I don't know why that is. That is a big question. Why do they show this upstream movement bias? But it's already shown up in multiple streams where we've done this kind of similar methods. So it really seems to be this consistent characteristic of movement in Geronopholis. But we get this upstream bias. So does that upstream bias somehow interact with the topography of these systems to influence genetic divergence within this species? Well, you might predict that to be the case. So if you think about variation in the topography of these headwater streams, you get some streams that have relatively shallow slopes and some that have relatively steep slopes. And we predicted that given this upstream bias, you might see more movement along streams in these shallow sloped streams and less in these more steep slope streams. And this prediction, I should mention too, is based both on that strong upstream bias, but also the fact that relatively few animals seem to move downstream. Because if you had lots of downstream movement, it would upset any pattern created by that change in upstream dispersal. But it seems like relatively few animals move downstream. So we might predict that as stream slope increases, gene flow along the stream or between sampling sites would decrease, leading to higher divergence in these steep slope streams. And for this work, we were working at Hubbard Brook, so we had multiple streams that we could use. And we simply set up sampling sites along these streams. So for each of these 9 streams, we had a downstream sampling site and an upstream sampling site. And in all cases, they were separated by 1 kilometer of stream length, so along the channel. But between those sampling sites, the slope of the stream could vary just depending on the topography of the drainage. And that's what this figure is showing you. So that figure has distance from the confluence along the x-axis and then elevation on the y for those 9 streams. So that's just a standardized elevational profile for each of the streams. And all that I want you to take away is that there's variation in the slope of those streams. So some are very steep here, and then some are more shallow across the 9. But in all cases, the sampling sites were separated by this standard distance of 1 kilometer. And we quantified slope, or variation in slope among those streams, as just change in elevation between the downstream and the upstream site And we use AFLPs, or amplified fragment length polymorphisms, to quantify genetic divergence or genetic distance between those two sites. So I'll show you some data points here in a second, but what you want to keep in mind is that data point represents the genetic distance between the downstream and the upstream site on an individual stream. And it seems so quaint to use AFLPs these days, like who uses AFLPs? But I did. But now we're doing SNPs and things like that. Okay, so this is just a cartoon of the sampling design. You have the fish-infested main stem down here, have our downstream sampling site, which was just 200 meters upstream of the confluence, a kilometer-long section, and then the upstream site here, and we repeated that on those 9 streams. And these are the results that we got. And remember, this is all within Hubbard Brook, looking at these 9 different streams. So each point here represents an individual stream and the genetic distance between the downstream and the upstream site. And what we saw is that as slope increased here along the x-axis, that's change in elevation, increasing to the right, you got higher divergence between the downstream and upstream sites. So it appears that this pattern of divergence was consistent with what we predicted based on the movement behavior. So given that upstream bias in movement, if you quantify gene flow or dispersal in a shallow slope stream, you get more dispersal, and in a steeply sloped stream, you get less dispersal. And it doesn't look like that downstream movement is kind of eroding that pattern. Okay, so that's AFLPs, lots of weird stuff going on with AFLPs. It would be nice to know if these patterns of genetic divergence had any phenotypic consequences for the species. And so to do that, we use the same simple methods of taking photos of the salamanders and quantifying morphology in the downstream and the upstream sites. In this case, we had to narrow our sites down to 6 streams. And we focused on trunk length here of the salamanders, which is pictured on my great cartoon of a salamander. So this is just the distance between the forelimbs and the hind limbs. And here again, we standardized our trunk length measurements based on snout vent length. So they're size-corrected measurements of trunk length. And there had been a couple papers that came out when we were working on this study that had suggested that trunk length was a really responsive variable, evolutionary responsive variable in salamanders based on some work in California. So that's why we focused on that characteristic. Went out to these 6 streams and then used the same basic method. So do we see patterns of phenotypic divergence between downstream and upstream sites that match the patterns of genetic divergence? And that's what's pictured here. So we have genetic distance here on the x-axis, that's between those 2 downstream and upstream sites, and phenotypic distance on the y-axis. So that's just a measurement of how different phenotypically in trunk length salamanders were along the streams. And what you see is that as genetic distance increased, so did phenotypic distance in trunk length. So it looks like these patterns of dispersal and gene flow that we had predicted based on the upstream movement bias have consequences for genetic divergence along streams at a relatively small scale. So we're only talking about 1 kilometer along the streams, but also for phenotypic divergence, kind of supporting this idea that there's something about these upland headwater areas that may promote divergence in intraspecifically, in this case, in this plethodonid salamander, but may also underlie those broader patterns of diversity we see in the family. Okay, so exciting. We get this evidence that the topography interacts with the dispersal pattern or this upstream bias to promote genetic and phenotypic divergence, but the big limitation here is that it's just 1 species, and it's hard to make a jump from this 1 species to those broader patterns of divergence or of diversity that we're seeing in the family. And because I work in the northern Appalachians, we don't have such great selection of plethodonid salamanders. So we have 4 species that occur at Hubbard Brook, whereas down in the southern Appalachians, you can find like 17 species along streams. So we don't have that many to work with, but we do have this guy. This is Eurycia bislineata, another plethodonid, the 2-line salamander. These guys are stream salamanders as well, so the larvae spend 1 to 3 years in the stream in the aquatic form, so with external gills. They then metamorphose and they move out into the riparian zone or even out into the forest where they hang out for multiple years and then return to the stream to breed. So relative to Geronophilus, this is a much more terrestrial adult phase. They're also smaller, much smaller than Geronophilus. So Geronophilus, I should have showed you, so they get to be about this big, whereas Eurythia is down here. And Geronophilus eats these guys too when they have the chance. But for Eurythia, I didn't have any information on how they move within streams. Thankfully, there were a couple studies back in the 70s and the early 80s that had looked at movement in Eurythia. And what those studies suggested is that at least within streams, so they didn't tell us much about how they move out in the larger landscape, but at least within streams, these guys seem to be really prone to downstream drift. So basically getting picked up by the current and carried downstream, especially in the larval stage. So you see, suggesting that they may show this opposite bias to what we saw in Geronophilus. Instead of an upstream bias, you have this downstream bias where the larvae especially are getting flushed downstream by the current. If that's true, then you might expect the opposite pattern of divergence with stream slope. So unlike in Geronophilus, here we'd expect that as slope increases, you get more turbulence, and higher flows in some of these streams, which may promote downstream drift, leading to higher rates of downstream dispersal, which sounds probably more active than it feels to a Eurythia larvae as you're getting flushed down the headwater stream, but higher rates of downstream movement and drift with increasing slope. So then you'd expect the opposite pattern in terms of patterns of genetic divergence. So dispersal and gene flow might increase with increasing stream slope. Again, same basic sampling design, go back to Hubbard Brook to 6 streams where we had Eurythia populations, choose these standardized sampling sites that were 1 kilometer along each stream, collect animals from the downstream site and the upstream site, and then look at the relationship between stream slope and genetic distances between each of those paired sites. And we see the opposite pattern to what we saw in Geronophilus. So in Eurythia, as slope increases here along the x-axis, you're getting a decrease in divergence. So in those high slope streams, appears to be more gene flow between those upstream and downstream sites than in the low slope or shallow slope streams. And also for Eurythia, we could go back and get those morphological measurements, again, focusing on trunk length in this case. And there we see the same relationship. So as genetic distance increases, remember in this case though, it's increasing as you move towards the more shallow slope streams, you also get matching patterns of phenotypic divergence, suggesting again that these proximal movement patterns that we quantified, or not we, but other people quantified in Eurythia, are scaling up to influence movement and gene flow along individual headwater streams, and then resulting in these patterns of phenotypic divergence at relatively small scales, so just 1 kilometer along these headwater streams in New Hampshire. And to me, it's useful to look at these 2 data sets on the same figure. We have 2 species, Geronophilus, the big pink one, and Eurythia, the little brown and black one. So ecologically similar stream salamanders occurring in the same streams in these headwater areas in New Hampshire. And yet they're showing these very different patterns of divergence along streams in the exact same streams. And the take-home message for me here is that it would be really hard if somebody were to just hand me these data and ask why are we getting these such different patterns of divergence in these such ecologically similar species. It would be really hard to interpret those without some information on their proximal movement patterns, so those direct data on dispersal. And that's the big take-home, is that having those data makes interpreting and predicting, even more importantly, these patterns much more scientifically sound. So in one species, in Geronophilus, you have topography promoting, steep topography is promoting divergence. In Eurythia, it seems to be acting the opposite, where topography actually impedes divergence among those populations. And I think that's really the take-home message here. I love thinking about all the complex consequences of dispersal in landscapes, in meta-populations, in meta-communities. But I think that this more bottom-up approach, so starting with these direct data on how animals move, even if it has to take place at a much finer scale that you're interested in and kind of in a scaled down study, I think that those direct data are super valuable because they, again, like I said at the beginning, they provide this empirical foundation for understanding those exciting larger scale landscape level consequences. So in these cases, simply knowing that we had this directional bias in dispersal allowed us to predict what might happen as the slope of the streams varied, and then go out and test that with these indirect indices, in this case, genetic distance between downstream and upstream sites. And so to put a little finer point on it, I think that we have enough models and theory about the consequences of dispersal, and it would be worthwhile taking all of that interest and directing it more to getting out there, turning over rocks, chasing after ground squirrels or whatever your selected species is, to try to quantify directly how are those animals moving and to build those empirical dispersal distributions, because I think they've just generally been undervalued in the literature. With that, I'd like to thank some people. First, some people who've supported me during the field season. Lorraine Turner, who runs the Merrill Brook site, basically. Kevin Evans, Don Busso, Ian Helm, and Jeff Wilson, who work at Hubbard Brook. My family, Jenny, Will, and Iris. And then these funding sources, the National Science Foundation, the Andrew W. Mellon Foundation, the Sweetwater Trust, and the University of Montana. And finally, thanks to you all for coming today. What Chris asked, which is a really good question, is, okay, so if increasing dispersal distance increases your survival and it increases your growth rate, then why on earth doesn't everybody go long distances, right? And the answer to that is really straightforward and hopefully not too disappointing, which is that there must be costs that we haven't been able to quantify. And so, yeah, it can't be a straightforward directional selection on long distance dispersal. We know there are costs, there are probably risks associated with the act of movement itself, whether it's exposure to predators, exposure to downstream drift by being picked up by the current. Those are out there. It turns out it's really hard to quantify that. So certainly there are animals that just disappear during dispersal, and we don't know what happens to them. And so when I look at those data, I don't think that's the end of the story. Like I said, I think it keeps the door open because it suggests, okay, even if there are all those risks out there and those costs, we still see these fitness benefits of increasing dispersal. So selection could still be acting, but is it not the complete picture of that? Yeah, great question.